Hello, everyone, and thank you for joining us today. Each time we come together, we're reminded how vital knowledge is in this journey. As parents and caregivers of children with neuroblastoma, we often face uncertainty and difficult decisions. Staying informed, understanding what's new and what's next, give us strength, perspective, and hope. Today, we have the honor of hosting Dr. Araz Marachelian, a pediatric oncologist at Children's Hospital of Los Angeles. Dr. Marachelian leads Neuroblastoma program at CHLA and serves as the medical director of the NANT Consortium, an international group of pediatric research institutions focused on early phase clinical trials for patients with relapsed or refractory neuroblastoma. In her role as medical director, Dr. Marachelian oversees trial development, patient safety, FDA applications, data monitoring, and publication review, ensuring that each step of the process moves us closer and safer and more effective treatments. Today, she'll share with us the latest updates from NANT, offering insights into current and upcoming clinical trials and what they might mean for our children and families. Dr. Marachelian, we are truly grateful for your time and dedication. Thank you for being with us today and for your hard work every day on behalf of children with neuroblastoma around the world.
Thank you so much, Annette. I'm really honored to be here today and be able to share with you what's happening with our trials. And hopefully this will be an interesting and interactive presentation. And I'm happy to take questions when we at the end. So this is not something you need to see. Here we go. So the first thing is I wanted to just introduce to the whole group what New Approaches to Neuroblastoma Therapy Consortium is and why it's structured the way it is. So it's a collaborative network accelerating new therapies for neuroblastoma. Of course, it brings together, you have leading children's cancer centers, you have scientists, and you have families, and all together to rapidly translate discoveries into clinical trials for children with relapsed refractory neuroblastoma. Although the focus is on relapsed refractory neuroblastoma, the reason for that is because the trials that are early phase, meaning coming from the laboratory to patients, really do focus on that patient population. But the idea is to always keep in mind what the phase 3 studies, perhaps for the upfront patients, and what are those needs, so that the appropriate studies are done in the relapse refractory population, but also how to affect the upfront treatment. So to give you an idea, this started as a collaboration in 1990 of 8 sites. There were a few trials that were being done. And then as it got a little more developed, there was a first grant that's called the Program Project Grant, which I'll talk about in a minute, that formed. And since then, there have been many trials. The trials have really focused on US and Canada in the beginning of NANT. But since then, we now have 18 sites, more than 1,400 patients enrolled. And now, as of in the 2020s, we have a collaboration with Europe. We did have a trial with Australia, but currently don't have a specific collaboration, although it is feasible. So here is what the map looks like. Really, I should get a Europe map here, but for the US, you can see the idea was to try and have sites across the country so that families don't have to travel as long to get to a big center. But these are specifically centers that treat a lot of neuroblastoma and have a scientific as well as patient focus on neuroblastoma. The little arrow is where I live, which is Los Angeles, and that's the main headquarters and operations, and our contracts, et cetera, are all based here at Children's LA. Our collaboration with sick kids has been around since in the early phases, but we did have our first trial, our lorlatinib trial, that included Institute Curie and Royal Marsden, and since then have formalized our European collaboration with Princess Maxima as being our essentially our European operations with the rest of our sites in Europe. Right now, in addition to these sites, there's a site in Germany that is being developed for our next trials. Just as a point, I want to make sure that, not all trials can be done internationally, and I think it really is dependent on, the agents and what we are able to achieve. But that is our goal, to try and have as many trials that are transatlantic. So you can see the group is a relatively big group. This is just the main people who were at a meeting for NANT. But the program project grant that I alluded to is a grant that put together groups of people to work together, essentially, for this unifying goal of turning discoveries into clinical trials. So there are multiple laboratories that are funded by this grant, and then it partially funds NANT. So really what I think about the program project grant, I think about it as our base to help us just get started. We still need plenty of other funding because this is a small part, but it does make the infrastructure really integrated and cross-disciplinary collaboration, et cetera. And so it allows for rapid translation of our trials. But it is a race, and it's a constant running around to be able to get these trials and improve access and hopefully have a curing potential. So I'll first start just to give you an idea on the type of trials that NANT has done and why. Trials are not just, I decided to do this one trial. There's a history around it, and when there's a direction of how we're moving. And so I think it might help us to just sort of think about them as blocks of types of studies at the moment, and then try and understand why these trials and how we're moving them forward. So, of course, I found this little picture of chemotherapy versus immunotherapy. This is a little while ago. There's no such thing as versus. It is an and. And we know that chemotherapy and immunotherapy has made a big difference in our patients in relapsed refractory disease. There have been great successes, but we also know there are lots of improvements that are needed. The neuroblastoma is smart, and it has a very immunosuppressive microenvironment all around it. I like this picture that, it is not, just the tumor itself. It's multiple cells that are talking to each other. And the more we learn about this talk to each other, the more we can maybe attack a specific crosstalk that is maybe not a good crosstalk for us. So we'll go through a few trials and how this was all done. So the trials that I'm going to talk about is, if we are trying to improve chemotherapy, what can we do? So number one, you can add cells. You can say, hey, neuroblastoma cells and the patient's immune cells are not good enough. And so we need to add certain types of cells. And one of these types of cells that you can add are these natural killer cells. And we'll talk about why in a minute. There's other ways to do this. Say, you know what, the cells are good, but we just need to give them a few more agents to make them work better. So these are a few agents that we can add potentially to make the cells that you have, your immune cells, work better. And then, of course, you can look at other immune molecules, so this crosstalk, and whether you can affect that crosstalk between the neuroblastoma cells and the immune cells rather than adding cells or adding, other cytokines. So first, let's talk about the NK cell trial. So what NK cells are cells that are a part of the innate immune system. They're the first ones that come when there's anything that happens. They're like your first soldiers, and they don't last a very long time, but they really inform the rest of the immune system, the T cells, the B cells, to pay attention to different things. And so the idea that they would be there for tumor and really detect tumor or fight the tumor first makes a lot of sense because they're the first things that also fight viral infections, et cetera, et cetera. So the idea then, well, do NK cells help anything? There were trials that were done to add NK cells to dinutuximab, and they were good killers of neuroblastoma. And we actually did a trial where we took patients' own NK cells, grew them. These are neuroblastoma patients, gave them back. And we realized, although we were able to grow them, they just weren't strong. And they were also, we couldn't grow them to the same number because these are neuroblastoma patients and their NK cell numbers are not huge. And so the idea came about looking at another way. Instead of taking them from the patients, could we now grow NK cells ourselves, have them ready, and give them to patients when they needed the treatment? And so that brought on the idea of, well, we can grow the cells, but what about if we grow them in an environment that's similar to what the tumor would release, which is it releases all this compounds to make you not pay attention, right? The NK cells to be not the great killers. And so the lab experiments added this thing called TGF-beta, which is an immunosuppressive thing to make your NK cells not being smart, and grow them in there so that they're used to it. I call this sort of imprinting, right? Because you're trying to understand which way to go and how to work well. And so when this NK cells had TGF-beta in their same environment and you gave them back with dinutuximab, they worked even better and they were better killers. Great. So now we need to translate this to patients. So that's where NANT 2102 come in. You have chemotherapy, you have immunotherapy, and you have NK cells. So the NK cells, how are they? You take a donor from a bone marrow registry, you get the type of NK cells that work the best, because all of us have different NK cells and some of them better than others. And then you can collect them, then you grow them and imprint them. And then eventually you have these supercharged allogeneic NK cells, allogeneic meaning it's from someone else. But because they're from someone else, people think, well, oh my goodness, well, is it like a transplant from someone else? And therefore there's all these rejection or graft versus host disease, things like that. That is not, you do not see this with NK cells. And the reason is because NK cells live a little bit, do their thing and go away. So they're not in there for a very, very long time. And because they're not in there for a very long time, what you have to do is give them every cycle. So in this case, you can see here the trial is irinotecan and temozolomide, which are our standard chemoimmunotherapy backbone. We have dinutuximab with its standard way giving, which is those 4 days. You have the chemo for 5 days, and a few days later, you come in once the chemo's out of the system, and it's avoiding a weekend, come in and give some NK cells back, and then you have your GM-CSF still helping your NK cells and everything else that's happening in your immune system. But what you have to do is you have to repeat this every cycle. And so far, patients have tolerated great and have been able to get the 6 cycles that the protocol has written for. And of course, the goal of the study is to look at what is the response rate when you do this and try to compare it to what we know the response rate might be for this patient population. This is an ongoing study. It was on hold for a little bit of time, but it's opening right back up. And now we have all the funding we need to get the study really to completion at this point. So that's NK cells. So I'm just going to touch upon two studies that are actually NANT affiliated in some way. So the first one is a true NANT affiliated study, which is this cytokine. If you remember when neuroblastoma, when antibody therapy was being given, there was also this thing called IL-2 that used to be given, which we really have gone away from because of toxicities and really not seeing major response differences. But the IL-15 superagonist is something that's sort of in that same group of cytokines. What it does is actually, this is an agent that's really gone forward in multiple myeloma and some leukemia, lymphoma. But what it does is that, again, expands and activates what we really want, the NK cells and your T cells that are, expanding. And it's given sub-Q, so it does have some local reactions in the skin, kind of like your GM-CSF. It is safe and really stimulates this immunity that you need. So the trial for this is a collaboration with St. Jude, where just like the other one, irinotecan temodar, the same way, but now you're giving this agent, which is ALT-803 or NO-803, on the sixth day. And then you have, following that, you have your GM-CSF. GM-CSF is just a little bit later so that you can see how you do with this sub-Q injection itself. So the way this study is written is actually it's using the humanized 1418K322A antibody. This is what people used to call the St. Jude antibody because it was the initial phases of the trial were all based at St. Jude. So because there's no real trial that's specifically in this patient population that looks at irinotecan temodar and humanized antibody and GM-CSF, this is a study that's randomizing. So half the patients will get the new agent and half the patients initially, and half the patients will get what standard chemoimmunotherapy. But there is a crossover, meaning if they have progressive disease, they're able to cross over to the other arm to get the NO-803. This trial is just in its early phases in activation, but plans to be opened at all the NANT sites, although being operated specifically from St. Jude. One of the good and important things of all clinical trials that we do is that we also are always trying to learn and understand why something works versus doesn't. So when I go to the biology aspect, there's a MIBG trial that's coming up, and all the patients on the St. Jude trial will also be participating in the MIBG trial. So there's some data, hopefully, that we'll be able to learn from based on this together. So I'm only going to talk about this old study that was run by me, which was a lenalidomide and dinutuximab trial. Just to say some of these numbers are really because of the year the idea came about. So 2011 is a long time ago. But the trial completed even during the time when dinutuximab was getting FDA approved. But what's cool is that this lenalidomide is an immunomodulator, and there was data to say that dinutuximab and lenalidomide together work better than dinutuximab alone. This is pre-chemoimmunotherapy era, and we did lots of really detailed analysis of both what's called the cytokines and the actual cells. So on the left, you can see the cytokines, and on the right, you can see these cells. And you can see certain things that are going up beautifully with the treatment all throughout the lenalidomide. And dinutuximab is given just these days, and you can see, for example, this IP10 specifically goes up with the dinutuximab, and these things, for example, you can see similarly. So really understanding from our patients how what's happening in their body. But similar to that, you can see, for example, these T cells with lenalidomide are just continually going up. And this is a good thing, right? We want active cells. So this was a really beautiful proof of concept with dinutuximab and lenalidomide. So because of the excitement of this data, and actually because there is now even better compound called iberdomide than lenalidomide, they're cousins essentially, or brothers. So there's actually a randomized phase 2 study that's going to open in Children's Oncology Group in the next year or so. And this is based on the same lenalidomide trial. But here it's chemoimmunotherapy, and the chemo is cyclophosphamide and topotecan and dinutuximab. So you can see how the work at NANT, for example, informed the COG trial, and that is what we really want to see. The reason this is a COG trial is because it's randomized, large study, and that way all patients at all COG centers could participate. So how else are we moving forward with immunotherapy? The next study that I'm going to tell you about is dealing with yet another part of the immune system. After this talk, you're going to become an immunologist, but you can see here there are multiple ways you can think about the tumor and the T cells and how they're interacting with each other, and also actually the NK cells. This is just a picture of a T cell. And the one that I'm going to focus on is this TIGIT that interacts with the tumor cell. So mainly tumors hide, right, by sending signals to turn off the immune cells. So it is just smart, and it's trying to be invisible. And if you can then, so this PD-1 and TIGIT are two of these sort of breaks that the tumor is putting in. So if you can go anti these things, then all of a sudden your body can see the tumor cell. So anti-PD-1 and anti-TIGIT therapies help the own immune system better recognize these tumors and so attack them. I'm calling this, it is not the official at all, so this is just for entertainment purposes, but really, it's taking off the tumor's disguise and releasing these breaks. So I'm talking about our little disguising of a Superman. So what is this study? This study, we have an anti-TIGIT compound, an anti-PD-1, PD-L1 compound that are going to be given. And then we have irinotecan Temodar. And here we have dinutuximab beta. And the reason this is dinutuximab beta is because this is a transatlantic collaboration where we're able to hopefully get both our European sites and our US sites to participate in this. And we thought that that would just be easier to have one antibody across both sites. So this study is not yet open, but we anticipate that the study will open in middle of 2026. And this will be another way to harness the immune system. So I'm going to now switch gears to MIBG. And MIBG has been around since the 80s, right? It's an analog of norepinephrine. Most of you know it from the fact that you get it for scans. But it's really because it's concentrated in the sympathetic nervous tissue, which is where neuroblastoma arises from. So the goal has been all along, great, this is a good targeted therapy, but how can we boost its response rate? I'm calling it DOG, targeted, the original targeted therapy. And I think about this as not the Superman, but really the Trojan horse, because it's being taken up by the cells, and now it's radioactive and killing the cells. So we'll talk about treat trials that are consecutive. They built on each other. So the first one was the addition of vorinostat. And again, we're talking about 2011 as an initial concept. Then we have a study that added dinutuximab and dinutuximab vorinostat. And we have a study which is called 2102, but actually going to open next year with the addition of all the vorinostat, the dinutux, and the vorinostat and dinutux arm. So how did this come about? So the early trials were, the early trial was the addition of vorinostat. Why? Because this is called an HDAC inhibitor. Mainly, it makes the cancer cells more open and active at the genetic level. When you give it with MIBG, it can increase the amount of radioactive MIBG that the cancer cells take up. And this is what's called a radiation sensitizer. And the study was randomized, multiple arms, looking at MIBG alone, MIBG with irinotecan and vincristine, and MIBG with vorinostat. And you can see here the response rate of the vorinostat was the better, the more arrows down, the more better response in our patients. And so this is the reason now MIBG is given with vorinostat when we give it standardly. Then the next study came about that's called NANT 1701, and that is just recently published. Mainly now we have MIBG and dinutuximab, two very active agents together to work together. And there's biology here that's interesting. But the one that I'm going to really focus on is this idea of what's called abscopal effect, where the radiated sites in a tumor then respond to in other sites when you have immunotherapy. So you radiate this one site, and then even though you're not radiating these other sites, if you have it with immunotherapy, you can get a better response. And this is a concept that has been seen in other tumors. And so this was the rationale for putting them together. But on top of it, then the idea of additionally, if we brought in vorinostat, in addition to what I talked about earlier, can also increase the tumor's GD2 expression. And if GD2 is there, that means the antibody can bind better. And so altogether seemed like a win-win. So you have this MIBG and vorinostat, and then you come in with dinutuximab on the different days. The first part of the study didn't have the vorinostat, and the second part of the study had the vorinostat. And so you can see here, this is the response rate on the patients. The different colors are what their responses were. But you can see where the response rate of this is different doses as we were going up. But the overall response rate was 46% in this part A, which is the one without the vorinostat. And then here is dinutuximab and vorinostat, and the response rate was 75%, which is very promising. Of course, it's a small number of patients, so it's clearly something that needs to be looked at pretty closely. And so then that is the reason we have this trial. That's a randomized 3-arm phase 2 study And I'm happy to say, even though the study is called 2102, it is going to open in 2026. And the reason of this delay was due to MIBG supplier issues. There was a company initially that was producing MIBG that was going to give us the study, MIBG, and unfortunately, that company went away, which is, the problem we have in pediatrics. And so and it's taken us a little bit of time to get our supplier and really come up with a trial that benefits both our supplier and our patient population and our science. So it took a little bit of time, but I'm happy to report that we are actively working on this trial now. We will definitely work on some funding for this trial, but we will be opening it for patients in 2026 and work on the rest of it. Hopefully, it will take quite a few years because it's 105 patients. So as long as we can get started, we will do so this year. So I'm going to switch over to biology for a minute. I know you as a parent who are listening and you're looking for, what are clinical trials that you can have your child on. This is really not, the focus of this specific presentation, although you can see the types of studies that are being done. And at any time point, of course, these trials, there are other trials that are in development, but not yet at a point where they're really launching. And so therefore, they're a little bit further. And so I'm unable to talk about all of them. So I'm going to pivot to the lorlatinib story, really to pivot into the biology of tumors. And so, this was what we ideally think about from lab to patients, right? The ALK mutation was identified. There were patients, we understood that there were 10 to 15 patients at diagnosis, percent of patients at diagnosis who had an ALK mutation, but in the relapsed refractory setting, this was a higher number. And since the lorlatinib was an agent that was being created for lung cancer, and it had better preclinical data than the usual ALK inhibitors. This trial in NANT, which was 1502, came about. But because this is a rare population, that was our first transatlantic collaboration. And you could see there were promising response rate, and now is in phase 3 trials in the US and hopefully getting there in Europe. And you can see the response rate, the colorful schema here of patients responding. Cohort A2 is older than 18 years of age patients, and those patients specifically seem to have a really even better response than our pediatrics. But the chemo plus lorlatinib cohort seem to have a similar to the adults. So why am I talking about this? This trial is now completed and the phase 3 trials is ongoing. So we really have to wait until we get the information from our phase 3 trials to see if upfront patients will benefit from lorlatinib upfront. And that study, ANBL 1531, run through the COG, just reopened for that arm. And it's going to take a little bit of time to get there. But in the meantime, it's really learning about our patients. So for example, you can see here, there's a, the red dots mean the patient had progressive disease. And at the red dots, the bigger the dot, the more the tumor that has ALK in it. This is liquid biopsy, and we're looking at liquid biopsy on the patients instead of biopsying them every month, looking at what is their tumor doing. And in the first group, you can see there are many of the patients who, when they have a red dot, they have a bigger ALK tumor. And then the patients who were not progressing, they didn't have any really of that. and their dots were not getting bigger. These are what we call ALK dependent, but there were patients who were ALK independent. So they were progressing, but there were other mutations that were coming about. And then there were some patients who had ALK in their tumor, but they didn't have ALK detectable in their blood. So there are different kinds of patients, and trying to understand these patients is how we would then figure out what our next steps are. There are things in the work for trying to figure out how to really target ALK. Currently, there's not a trial in the relapse refractory setting that's open for that, but certainly something we are working on at the moment. How does this relate to what we are doing? What's really important is that every time a patient is getting a disease evaluation is to get blood from the patient so that we can keep it and store it and then look at it and look at liquid biopsy, etc. We have a bank that's a rich repository of specimens for, with clinical annotation, meaning we know what the patients were, how much disease they had, what was going on with them, and so that you could sort of inform what kind of samples to give to different people. And that has been a very productive collaboration. And you can see here, there was a study that was done again to say neuroblastoma is not one thing. There are different types of tumors within it. And you can see the relationship of different genes to different genes. This is not just random. When you have a blue dot, it means the relationship is high. And, if you have it's the other way around. And you can see different tumors have a different relationship with each other, with the different genes. And when you understand that, you will probably be able to understand how to overcome some of the problems. So, for example, if I were to look at ALK here and its relationship with different things, you can see how that works, and then you can think about how to now overcome the problems with ALK. So going forward, what is really important for us, since the science of neuroblastoma and just science overall has really advanced, is for us to have a consistent way of evaluating all our patients at all times. So at all times, meaning every time they get a scan and a bone marrow, etc., we're putting together a bunch of assays that we're all going to be doing consistently, whether in the US or in Europe, so that we can look at what we call tumor evolution. The tumor is evolving, is changing, and we need to understand it. So a lot of this tumor evolution that I talked about earlier, the liquid biopsy makes lots and lots of sense, right? It's easy, it's blood, we can look at it, we can really understand what's happening in the tumor. This is, of course, happening, outside of studies as well in our relapse refractory population, but in this study, it's being really studied all together for the patients who are on these clinical trials. Looking at their biopsy of their tumor, sometimes it's a new biopsy and sometimes it's old, and looking at other things like tumor expression and immune profiling, and really storing things for also, future studies, because we know the technology is changing so fast. This study is called MIBG. It has a number, 2301, I think, but don't quote me on that because I forgot the number, because it is coming up again next year, but it's a molecular and immunologic guided high-risk therapy, neuroblastoma therapy. But what we will be giving the liquid biopsy and the genomic information back to the patients, so that their doctors can also look at that in the context of the trials. The laboratory studies that are happening in Europe are in parallel to the US, but are, of course, done at the different parts and not sending blood and tumor over to across the continent. The goal ultimately for something like this is to have a trial that you potentially, like let's say a lorlatinib, that's biomarker-driven trial, and then we can do data analysis, and then we can have a novel biology, then we have another study, and that's the concept. What we really need are more agents that we have access to, and that is work that is ongoing in terms of advocacy from the community of researchers and families. Within the study, we're also trying to understand patient-reported outcomes. So we are looking at some of these questions and to understand what is the experience of families on these clinical trials, and what are the main issues, whether it's financial, travel, et cetera, that are in the works. So we're doing this across all the studies in MIBG because it makes more sense to pull it all together rather than study it individually under each study. So all patients, once the study opens, and I do think that this study will open hopefully by the end of this year in the US and probably a little bit later in Europe, but once the study opens, all patients who are going on a NANT study will be enrolling on this as well and get all the results. And I never stop talking about neuroblastoma without talking about response, because ultimately for us to select what is a good agent, it is important to know how to evaluate neuroblastoma. I think you all have been in situations where maybe your patient feels better and maybe the scan is not really reflecting that, or there's disease that doesn't make them go on trial. All kinds of permutations. Do you need a bone marrow at every evaluation? There are lots of questions to be asked in response. Part of this work, I'm proud to say, led to the new response criteria. That's the international response criteria. But this is ongoing at the moment, and we're trying to understand how to evaluate it better, incorporating MIBG avidity for lesions that maybe are what we call cold tumors, looking at other imaging modalities, and the most important thing here is that you only can do this when you have this collaborative effort over multiple studies. So in closing, really where we're going is our goal is to expand access to innovative therapies. I know that it feels slow, and some of this is because we don't always have agents, even though we are very interested in this advocacy and collaboration, whether it's within the scientific community as well as with families, is really important. And integrating precision medicine, I know precision medicine is a beautiful word, but reality is we need to see how many patients can actually benefit from all these studies, but also remember that some of this is for our future, because we do know that we're going to have to learn more as time goes on. So every sample teaches us something new, whether the patient has progressive disease or not. And what I just said earlier, but it's really uniting this research, advocacy, industry, and regulatory partners to accelerate our cures. But ultimately, we're in a race of supply of agents, cost of agents, and tumor evolution. And we have to balance this while we try to get these studies up and going and available for everyone. I think I touched upon all this, but our role in global research, we do, neuroblastoma is rare enough that we don't collaborate. We're not going to be making progress. And I think coming through it with an idea that we don't always know the answers and our assumptions are not always true. And what is really important is to be scientists rather than just believers of things, saying we need to learn it, we need to study it and learn about it before we can go forward. And so these are where the crosstalk is really important. And obviously, we want to avoid duplication and harmonizing standards. And so this work across multiple institutions as well as transatlantic is very helpful. And every research part of the process is a time when our advocates can be involved in. So we actually have a NANT Parent Advisory Committee that we're very thankful for, as well as the whole research community and patients and families who trust us with their children to be able to give them access to therapies, but also learn from them. And that is all I have to say. Thank you very much. I'm happy to take any questions. I think this is the slide deck for information for you for sharing. Right. So maybe I'll take a minute and get to our Q&A and we can see how we can answer some of these questions. So our first question is about MIBG supply. It's been an issue over the years. Do we anticipate more MIBG supplier issues in the future? And I think I sense that, this is an important topic for both, at this point, I'm talking about Europe versus the US, but, it's an issue across the globe. And it is something that the neuroblastoma community is working really hard with industry to make sure that these agents are available. And some of that has to do with us helping design trials that could also help them in their regulatory pathway. Because ultimately, we want agents like this to be FDA approved so that it is not an issue of do we have access or not. And so that is an active part of our work at the moment, and actually looking at, for example, our trial may be used for future regulatory purposes, and hopefully that will solve many of our problems. At this point, I'm assured that we are going to have supply for the current MIBG study in the US, but I think over in Europe, for example, GE, I believe, makes MIBG, and that is in progress. So hopefully we can have other MIBG trials once we get more assured supply. So in terms of the next question about if MIBG is for first diagnosis or relapse refractory, there was actually a trial that was ANVL 1531 that studied MIBG in the upfront setting for prior to relapse. That trial completed enrollment. So patients currently who are upfront and not relapse and refractory in the US are not receiving MIBG because there isn't another trial right now. The results of that trial, because it was randomized, should come up in next year. So I anticipate by perhaps fall of next year for the results to come out. So currently, MIBG is really for the relapse refractory population, and that's the population that we know so far have been able to respond and benefit from it. So ATRX mutations and neuroblastoma, that's chronic, although lots of ATRX and mutations in neuroblastoma, even in some patients who have less than chronic, probably neuroblastoma. Currently, there aren't any trials open. There are some in development. And so keep an eye on those, and then we'll see. There was even a trial looking at perhaps combination with ATRX as well as radiation, but currently not anything that's open specifically for it. The next question was about our current political landscape in the US, and NIH cuts, et cetera. Obviously, you can realize that it is a difficult time in the US for researchers. We are currently not specifically impacted with the program project grant, but of course, there are always going to be reapplications, and there are many other applications as well that are going in. And so, of course, I think us saying that we are not impacted would be probably not a full correct statement, but our PPG funding right now, as of now, has not been impacted. How do I foresee this for the future? I think it's all about just like any investment portfolio, one has to be diversified in their portfolio. We really depend on our program project grant as a base to allow for being creative and being able to quickly turn around without having all our funding in place to be able to start a trial. But we just have to be really smart, continue to advocate, and continue to then try to get more funding. And I think the next question is AI. I know, I think there was President Trump signed something for AI and extra funding for AI to assist in clinical trials. And we've actually been talking about this and trying to see if any of this could really be helpful for us. I think it is still in very early stages, and I think right now I don't see AI as changing drastically anything in our trials. And that is not because we are not interested in adding AI, but really that is not where we need more funding at the moment. Although, I have seen some industry studies where they've tried to look at many, many tumors and looking at preclinically, whether, AI could then come up with a molecule that may be more helpful that we don't know about, things like that. I think that is possible, but, right now, pretty early, and hopefully AI will be more than just theoretical. And then I think this may be the last question, and thank you. I think what therapies am I really excited about for relapsed refractory neuroblastoma? I think, I'm one of those people who wants to say I don't want to have a favorite child in some way, because I feel like our patients need all kinds of treatments. And it is hard to anticipate before which one is ultimately going to be the best one for them. But I think some of these chemoimmunotherapy platforms and some of these new agents, I sit on some, adult cancer review committees, and I listen in or evaluate some of the new trials. And there's so much happening in immunotherapy in adults. So I expect a lot of that to come down to us. No, we're not down, we're high. But either way, I do think that these agents are going to be very exciting. I know there's a lot of excitement with CAR T cells and things like that, which we didn't talk about today. But I actually think there's a lot of excitement with other things like what's called ADCs, which are, these antibodies together with some chemo payloads. There are all kinds of things that are happening that are really making a difference in adult tumors. So I think keeping our eyes open for all those and working with industry partners to get them in pediatrics for the right patients, I think is the way to go. But we shall see. We will continue to work and find out, and that's where we have to come back and see for which patient is one better or the other. But I think we have a long way before we can make that specifically, AI, this is it. This is your answer, and we oncologists can go out and, have some vacations. Just kidding. But I think we're a long way from actually answering which one is ultimately the better thing for our patients. But for each patient, each individual physician with the knowledge can hopefully help to come up with that answer for the patient and what trial is probably best for them. And I think those are all the questions that I can see in the chat. Let me know if you have any other questions.
Thank you so much, Dr. Marachelian, for sharing your time, your expertise, and your clarity with us today. It was amazing. Thank you. Your work and your team at NANT are leading, gives the families like ours hope that is grounded with science, collaboration, vision, and for better, safer treatments for children with neuroblastoma. So really, really thank you for that. To everyone joined us today, thank you for being part of our growing community. We know how heavy this journey can be, and we hope these sessions help you to be more informed and well connected. As we look forward, we also preparing for the International Neuroblastoma Awareness Day on December 5th this year. We're proud to have support from institutions from all over the world, and we're building truly meaningful programs, one that touches on the topics that you frequently ask the most. This is your day, a day that belongs to our entire community. We invite you all to join us, collaborate, and raise awareness, because together we can make our voices stronger and remind every family that we are not alone in this journey. Thank you again. And our next session will be from our German group, which is Dr. Holger Lode, who will talk about immunotherapy and the new clinical trials they're having. Thank you so much for joining us today, and we're looking for our next session.